For my experiment, I initially started with Nigella sativa seeds, but then I had to change it to the brown fava beans because the Nigella sativa seeds started to mold and there was no growth with them. So for my experiment, I initially started with the black Nigella sativa seeds, and for the first treatment, I put around 20 of the seeds in a Ziploc with a damp paper towel and I taped it to the window in my room. And for the second treatment, I put the same amount of seeds in a Ziploc, but I dampened the paper towel with a tablespoon of sugar water. So I put a tablespoon of sugar in water and I mixed it around, and then I used that to dampen the paper towel. And I also taped it on the window in my room next to the first treatment. And this is the same thing I did for the brown fava beans as well. So this is what they look like initially. They're really small, which is why I used around 20. And my hypothesis was that the seeds in treatment 2 would grow faster than the seeds in treatment 1 because I assumed that because of the sugar, the nutrients would speed up the growth process. And this was also my hypothesis for the brown fava beans. So my experimental variables were that my control group was the beans and the seeds with only water that was used for the paper towel, just plain water, which was treatment 1. And my independent variable for experiment 1 was the Nigella sativa seeds, and for experiment 2 it was the brown fava beans. And my dependent variable was the amount of growth. And some of my control variables included the room temperature and the water temperature and the exposure, because they were all taped right next to each other on the same spot on the window in my room. So they had the same exposure to sunlight, and my room's temperature is also the same, and the water used was also all just at the same room temperature. And the amount I used to damp the paper towels was also very similar. So I took a picture of them every day to see if there would be any growth. So I took a lot of pictures, but by the end of week 2, my treatment 2 completely molded, if you can see in the pictures. So I threw it away, but I kept treatment 1. And then I started the brown fava beans. So this is what they look like. And then I had my 2 treatments, the one with the dampened paper towel with water, and then the second one with the sugar water. And then you could see that they're taped to my window, and that's what it looked like from outside of my window. And so for the results of the fava beans, there was no growth at all in either of the treatments. Actually, treatment 1 molded completely, which is surprising because that's what happened with treatment 2, the one with the sugar water for the Nigella sativa seeds. And then this was treatment 2. They kind of shrunk a little bit, and they also molded a little bit, but not as much as treatment 1 did. So there was no growth at all. And then for the treatment 1 that was left over for the Nigella sativa black seeds, one of the seeds actually sprouted 1 millimeter out of the 20 that were in the bag. And this was after 6 weeks because I continued it along with the brown fava beans. And yeah, thank you.